today. So first we'll be going through an introduction on DDDPlus, the background of the dissolution process, theory, and program features within DDDPlus. After that, we'll be going into the new features that we're providing in the new version 5.0, the new models and apparatus that are available for simulation, and more importantly, the ADMET predictor integration that we've included in DDDPlus 5. Finally, we'll go through some case studies and show the link between GastroPlus and DDDPlus by doing some IVIVC and dosage form design. Then after we have designed the dosage form, we'll use it to perform virtual trials that show dissolution specifications and then the impact on bioequivalence within GastroPlus. So how can simulation software be used? One important aspect is dissolution method development. How can I design my in vitro experiment to correlate with the in vivo release? So on the left, we have a couple in vivo dissolution curves. And what we're really trying to accomplish on the right is use our DDDPlus models to see if we can't build an in vitro method that's one-to-one correlating with the in vivo situation. Also, formulation design. How do I modify my formulation to produce an in vitro dissolution rate that meets my target? So once I have the dissolution method developed, I can ask what-if questions. What if my particle size changes? What if I change the excipient levels, disintegrant amount, that kind of simulation. So establishing dissolution specifications. Here on the left, I'm showing a plot of a virtual trial where we simulated the variability around HPMC content in a controlled release dosage form. Using the virtual trial functionality within DDDPlus, we're able to simulate the likely range of the dissolution, both the high and low dissolution rates that we would expect from natural variability in our formulation. We can then assess the impact in vivo using GastroPlus. Using GastroPlus, we can predict the plasma concentration time profiles for both the high dissolution rate and the low dissolution rate, and see if these are bioequivalent and or we need to tighten our dissolution specifications or we can leave them loose. This is another important topic that we'll be going over today in our examples. So what is DDDPlus? It's a state-of-the-art formulation simulation computer program that contains equation to account for all the aspects of dissolution. So the dissolution rate of the active pharmaceutical ingredient and all excipients are accounted for within the DDDPlus model, provided you have the required inputs for solubility and etc. We can account for multiple particle size distributions for both the API and all the excipients in the formulation. So not only can you specify mean particle size and then the spread of that distribution, but you can also use your experimental data and or fit distributions to your experimental data to do predictions of release rate. We have a variety of dosage form models that are available for simulation within DDDPlus, from IR products, both powder, capsule, tablets, DR products, coated tablets, and then CR products, matrix tablets, and new to version 5 is the bilayer tablet dosage form. Within DDDPlus, we have the ability to simulate all the nonlinear pH equations and calculations for both the API and excipients. We can do a dynamic microclimate pH calculation at the surface of these particles and also a calculation of the pH in the bulk fluid itself. This takes into account all the buffers in the system, the compositions of acids, bases, and salt equivalents. We have various selections of USP apparatus, and new to this version, version 5, the p-ion microdisc profiler that allow you to change the experimental conditions, the volumes, the instrument speeds, and see how it impacts your dissolution. We also allow for multiple experimental phases so that you can design your experiment to be more one-to-one correlating with in vivo situations. So if you wanted to do, let's say, a gastric transfer to intestinal buffer type dissolution experiment, we allow that flexibility within DDDPlus. And then finally, we can also model things like micelle-facilitated fusion through the incorporation of surfactants into the media. So we have beefed up our bio-relevant solubility features within DDDPlus in version 5. And we also have the ability to model surfactants such as SLS. The inputs that you would need to define a DDDPlus model, first for the formulation, you would need to know something about the compression force of your tablet, the tablet dimensions or diameter. Maybe you have a measured in vitro disintegration time. You may have measurements on the tablet porosity. And if for a controlled release formulation, you may have the matrix dimensions or the tablet size. And that will determine what the area of the dosage form is for release. For each ingredient in the formulation, you're going to need to know the physicochemical properties, whether it's the API, wetting agent, disintegrant, or other. You're going to need to know the molecular weight, diffusion coefficient, pKa's, solubility at a reference pH, the solubility factor, which is the ratio between the charged solubility and the neutral form solubility, the particle size distribution of your different ingredients. We've added the ADMET predictor module into DDDPlus version 5 that allows you to define these simply with structure using our QSAR models. We don't have in vitro data available. For the experiment, we have multiple different vessel types that you can model, so you're going to need to define that, the speed at which the experiment is run at, or the RPM, the media volume, initial pH, whatever buffer formulations they have, and we have a built-in database of many of the different buffers that are used commonly. Surfactants, and then different experimental phases. If you have a transfer experiment from pH 1.2 to 6.5, for instance, you would define that for your experiment. Formulation models. So within DDDPlus, we've added some new formulation models to our existing models for immediate release. We have powder formulations, and this is just a neat powder of API, and you can also have physical mixtures of other excipients in the powder dosage form. Tablet is an IR tablet model where we calculate the disintegration with a disintegration model. And then after the tablet disintegrates, the primary particles will dissolve based on several dissolution models, which we will get to later on in this presentation. Capsule is similar, except for now we would be defining the capsule disintegration time. And then new to version 5, we have bead coating formulations. This is crystalline API being released from a polymer coating or binder that is spray layered onto a bead core. Also new within DDDPlus 5, we have included delayed release formulations. So in this case, if you had a coated tablet with a pH-triggered soluble or insoluble coating, let's say something like methacrylic acid or HPMC, and you had a transfer experiment from pH 1.2 to 6.5, we would be able to model this and simulate the dissolution of the coating and the drug diffusion through that coating until it is completely dissolved, and then the tablet will disintegrate and release drug as a normal IR tablet. Finally, for the controlled release formulations, we have matrix tablet formulations for both swellable and non-swellable matrices. And then new to this version, version 5.0, is the bilayer tablet model, which incorporates the polymer matrix swellable model with the IR tablet model, modeling those two simultaneously to predict IR-CR formulations. The DDDPlus experimental apparatus options are obviously the USP1 basket and USP2 paddle, which are the workhorse of many dissolution experiments and dissolution testing. But we also offer the flow through USP4 in open or closed loop mode. And new to this version, people are doing dissolution experiments in much smaller volumes and much more rapid throughput types dissolution screening. And we've included the Pion microdisc profiler as a new feature into DDDPlus 5. The velocity that's calculated based on the RPM is based on fluid dynamics simulations. And so all that is built into DDDPlus 5. There's also the opportunity to define your experimental design. So redesigning in vitro experiments can help when you have an in vitro, in vivo correlation that's not quite achieved or is not one-to-one. So if you need to modify your dissolution experiment such that you get a more similar situation to the in vivo conditions, you can do that by defining phases within DDDPlus. This lets you optimize the in vitro experimental conditions to produce an output desired dissolution profile similar to the in vivo dissolution profile. Available is the instrument speed, medium volume, and pH for each phase. You will define the end time of each phase. And then with these features, you can mimic, but not exactly reproduce the USP Apparatus 3 type experiment. The outputs that you can obtain after building these DDDPlus models are, you can predict the dissolution profile for all formulation ingredients. We're not simply just looking at the API within DDDPlus, but we're looking at all of the excipients as well. So on the top left, we have a graph of a CR dosage form where we're predicting the amount of drug released, the polymer dissolved, and the lactose that is dissolved from that matrix of adanazolam. We can also calculate dynamic changes in media pH and microclimate pH versus time, as shown on the right, for that same formulation. And then if we want to do what-if analysis, we have a parameter sensitivity analysis function that allows you to see what the sensitivity is for any input variable within the DDDPlus model. So let's say we wanted to see what the effect of particle size is, you can vary it from, let's say, 1 micron to 100 micron, and then plot it on a spider plot and view the output. Additionally, we provide an F1 and F2 statistic calculator that lets you determine how similar or different your formulations are to a reference. And then finally, we have the ability to do virtual trials. So if you have a base formulation and you want to see the effect of your process variability, maybe your the variability of the pH in your system, the stirring rate, the volume in your dissolution apparatus, you can do that. And also you can take a look at content uniformity issues, changes in particle size, doing stochastic simulations, and then model that variability. This is going to help you set dissolution specifications, and then we'll show how these variabilities can be used to predict PK within GastroPlus. So we're quickly going to go through the main dissolution model within GastroPlus, which is the mass transfer model. We don't have the time to go through every single theoretical equation within DDDPlus, so we're just going to go through the main, sort of workhorse equations, and then later on when we get to CR dosage forms, we'll be talking about those equations as well. But DDDPlus, the default model is based on the mass transfer equation, which assumes forced convection around a spherical particle. The important variables to note here are the mass transfer coefficient, which really includes the hydrodynamics of the system, the velocity, viscosity, the diffusion coefficient, and then the particle properties like the particle radius and the density. Other important variables to note are the hydrodynamic boundary layer for the particle, sort of the stagnant fluid layer that sits around outside the particle, the solubility of the ingredient. So knowing the pH solubility profile is key in predicting the dissolution rate. And then also the mass of undissolved material. And that is all used to calculate the change in mass versus time, or the dissolution rate. Looking a little bit more in detail on the mass transfer coefficient, we use the standard Reynolds number, Schmidt number correlation to calculate the Sherwood number, which is used to then back out the mass transfer coefficient. The assumption here is that we are using the slip velocity, or the velocity between the media and the particle, to define the mass transfer coefficient. Fluid velocities for each instrument and speed have been estimated using computational fluid dynamics. Again, we have more equations. We provide the Johnson model for spherical and cylindrical particles. We provide the Nernst-Bruner equation and an intrinsic dissolution rate calculation as well, models that are built into DDDPlus, along with the disintegration models, the models for wetting, solubility enhancement due to surfactants. All those complications are also within DDDPlus. We simply don't have the time to go through every single one of the equations, but keep in mind that those are all available and used within DDDPlus. So selected new features within DDDPlus 5. I'm not going to go through the whole list right now in detail, but just point out a few main details. The ADMET predictor integration is key in predicting the in vitro properties of your API, and also can be used to predict the properties of excipients, provided they're of low molecular weight and they're drug-like molecules. Again, we have the new formulations that we've added within DDDPlus 5, and we have some new facilities to improve the workflow for biorelevant solubility, surfactant solubility. We've added some new features for tablet disintegration, and then some new output options for our parameter sensitivity analysis. The ADMET predictor module is a module that provides QSAR modeling that allows you to simply import a structure using a SMILES string, a mol file, an SDF file. You can load these structure files in, and then it will automatically predict all the physicochemical properties based on our QSAR models, and populate a DDDPlus database that contains estimates for the pKa's of your molecule, the full aqueous solubility versus pH profile, and then the biorelevant solubility, so the gastric solubility, the FASF solubility, and the FESF solubility. With those solubilities, we can calculate the solubilization ratio, or the impact of bile salts in the media. Additionally, the log D or log P versus pH profile is predicted, and then diffusion coefficients in water. We've given you more, increased flexibility to add more in vitro data to DDDPlus 5. In the case of surfactant solubility, we've added the ability to calculate CMC and solubilization enhancement factor for a surfactant drug combination using a data fitting tool, rather than previous versions of DDDPlus where you had to optimize the parameter using an optimization module. Here you can use in vitro data to define the inputs of surfactants in your model, rather than just using some fitting routine. So you can really utilize what in vitro data you have to directly input into the models to calculate the solubility enhancement of surfactants. Additionally, we've synchronized the bile salt treatment in DDDPlus so that it provides the same easy-to-use tools as GastroPlus to calculate the theoretical solubilization ratio. We've added the ability to utilize in vitro FESF and FASF solubility data and automatically calculate solubilization ratio. We've also added the ability to have a user-defined biorelevant media. So if you have some sort of solution that's slightly different than FASIF or FESIF, you can add that in as well as the solubility in that media and calculate the solubilization ratio. We also provide the theoretical solubilization ratio calculation that will use the log P of the molecule to define the solubilization ratio. We've added some new features for the tablet compression. So we have the Reimagine-Duckworth equation that describes the porosity of multi-component tablet mixtures. If you were measuring tensile strength of your tablets and you have information regarding the properties of your excipients, you can determine the porosity of the multi-component mixture using the literature, derived formulas. Also, you can directly input porosity now as a standard input. Another new feature for disintegration is rather than using our theoretical model to predict disintegration, you can simply use a in vitro disintegration tester value and enter that value into the tablet disintegration time. And that'll predict the time to 90% released of the API and excipients from the tablet. After that, then the dissolution models are going to take over and calculate the dissolution of those particles. And then again, as I mentioned earlier, new functionality for the parameter sensitivity analysis that allows you to do two-dimensional PSA and plot the results in 3D, which is sort of a pseudo DOE approach that allows you to look at the whole space and determine where your sensitivities are between two variables, in this case, solubility and mean radius. So now to our case studies. So for our case study, we're going to be going through a model-based formulation design. And this is going to be using the functionality within GastroPlus to define IVIVC correlations, and then using those IVIVC correlations to back out what the in vitro dissolution profile is for a target PK profile, and then designing a dissolution or designing a formulation and the amount of polymer in the formulation to hit that target profile, in vitro profile, and then in vivo concentration time profile. So this is really the synergy we're talking about within GastroPlus. So in the first part of the example, I'm just going to be going through the IVIVC and showing how to perform the IVIVC and then design the formulation. Once I've defined the formulation, then we'll go in and simulate the dissolution variability around that formulation and determine what the BE limits would be in GastroPlus. For right now, we'll go through the IVIVC and the dosage form design. So this slide is a little busy, but it is the high-level overview of the steps that you need to take in designing a formulation using GastroPlus and DDDPlus. So first off, you need some in vivo data to define an IVIVC relationship. So you need both the in vitro dissolution and in vivo plasma concentration profile for one or more formulations. And then you can generate an IVIVC, or a correlation between the in vivo dissolution and the in vitro dissolution. Then you need to define a target PK profile for the dosage form you'd like to design. So in this case, this profile is what we're going to use, this human CR fast, as our target profile that we're trying to design a formulation for. So once we have the target PK profile, we can deconvolute the in vivo dissolution using GastroPlus. So within GastroPlus, you can use the optimization module and the Weibull function to calculate the in vivo release profile, which in this case is this blue curve up above. And then that gives you the fit of your target PK profile. Once you have this in vitro dissolution rate, you can use the IVIVC that's been developed, and then you can use that formula to then calculate the in vitro dissolution that this in vivo dissolution corresponds to. So using the IVIVC, we can rearrange the equation using the quadratic formula, then predict the in vitro dissolution that corresponds with that in vivo dissolution that we deconvoluted. Once we have the, and that's these red points here in this graph in the bottom right. Once we have that predicted in vitro target dissolution profile that we can then use DDDPlus to optimize our ingredient amounts to best fit that dissolution curve. And then that is going to be our optimal formulation, which we bring forward and test in an in vivo study to see if we hit our target. So that's the overall, that's the overview of what we're trying to accomplish here in this first step. So to go into a little bit more detail, what is an in vitro in vivo correlation or an IVIVC? The working definition is that it is a predictive mathematical treatment describing a relationship between an in vitro property of a dosage form, in this case the dissolution rate, and a relevant in vivo response, the plasma concentration versus time data. And this comes from the FDA guidance. So, you know, traditional deconvolution, you're simply going to be determining the functionality between your in vitro dissolution rate and the systemic bioavailability, or how much drug entered systemic circulation. This doesn't tell you anything about what the in vivo dissolution rate is, you know, what processes happened in between the drug being dosed and it entering the blood. What we really want to know is what our in vitro dissolution rate is versus our in vivo dissolution rate, so that we can design a dosage form that will hit the correct PK profile. And the way we do that in GastroPlus is by using the mechanistic deconvolution method. So rather than deconvoluting the in vivo input rate and relating it to the in vitro dissolution, we use the full ACAP model within GastroPlus that has all the physics. So the permeation into the enterocytes, the saturable metabolism, and or any transport mechanisms that are present in the gut wall. Once the drug gets into the portal vein, then of course it can be extracted with first pass extraction, saturable or linear clearance in the liver, and then finally then the drug will enter systemic circulation. So by using this mechanistic deconvolution, where we, instead of looking at the systemic circulation, we're using the model to back out what the in vivo dissolution rate is and correlating that to our in vitro dissolution rate. So that's really the power of GastroPlus and the IVIVC methods that we have within GastroPlus, is it allows you to, instead of just looking at systemic circulation, to look at the in vivo dissolution rate. So the data set for our example comes from this paper from Dutta, and this is the in vivo and in vitro data in that paper. And to show the power, we're not going to be using all the in vivo and in vitro data in this example. We're just going to be using a small subset of it to show that, you know, you can do these predictions if you only have one or two formulations within in vitro and in vivo data. It's still possible to use these functionalities to then back predict an in vitro dissolution rate and a formulation in DDDPlus that will hit a target PK profile. So the questions we want to answer here are: can we create an IVIVC for the slow and in this case it'll be the fast and the slow formula, slightly fast and the slow formulations? Then can we determine the in vivo dissolution rate of the fast formulation using the deconvolution within GastroPlus? Then can we utilize the IVIVC that we've developed to then convert that in vivo dissolution rate for the fast formulation into an in vitro dissolution rate, and then predict the formulation composition that would hit that in vitro dissolution rate. So the first step in this process is to form the IVIVC. So selecting the slightly fast and slow formulation, we're able to run the mechanistic absorption model within GastroPlus to deconvolute the in vitro dissolution rate versus the in vivo dissolution rate and form a correlation, in this order, a second order polynomial function that describes the in vitro dissolution versus the in vivo dissolution rate. GastroPlus also provides the facilities to then see how well your IVIVC fits the data. In this case, the mean absolute prediction error was about 10%. For AUC, it was much better. It was about, you know, close to, you know, only a couple percent difference in this case. But the status window will display those validation statistics, and then you can select any combination of drug records that you have in vitro data available for to predict using the IVIVC module what the plasma concentration versus time profile is, shown here on the right. So back to our you know, overall steps that we're performing in this model-based formulation design. We showed you the first step, which was to generate the IVIVC. Now we define a target PK profile. So in this case, we're assuming we don't have any in vitro data for the human CR fast formulation, but we do have the pharmacokinetic profile, and we're going to be using that as our target profile. Within GastroPlus, I can then calculate what the in vivo dissolution rate is using the optimization module and the Weibull function. That gives a fit to the data shown here. Once I have that in vivo dissolution profile, I use that polynomial correlation between the in vitro and in vivo dissolution rate that I had developed on the previous slides. But instead of predicting in vivo dissolution rate, Y, I want to predict the in vitro dissolution rate for the human CR fast formulation. So I'm going to invert this formula using the quadratic formula to predict the in vitro dissolution rate. Doing so, I predict these data points over here on the right. And in this case, I'm outputting points that correspond with the data points that were sampled in their in vitro experiment. So they sampled at 60 minutes, 2 hours, 3 hours, and I think this is 6 or 7 hours. I can't remember exactly. But these are the predicted in vitro data points using the IVIVC to hit this target PK profile. So that's step 2, 3, and 4 in the process. So now that we've defined this in vitro profile, we're going to use DDDPlus to design a formulation to hit that profile. So now we're going to go into the a little bit of background on DDDPlus, but first let's look quickly at the formulations that were in the paper from Dutta. And the question, again, we're asking is, could the authors have predicted what formulation composition would have hit the fast release target in biostudy a priori? So could they have predicted this as their target PK profile? Could they have predicted the amount of polymer in the formulation that they needed to hit that PK profile? And then can we build a DDDPlus model for that one of the other formulations where we did have in vitro data and use it to design and optimize the excipient levels for that formulation? To do this, we're going to be using the power law release model that's available within DDDPlus for swellable matrix tablets. This model is based on Corsimire pepis. We've made some modifications to account for multiple polymers. If you're doing a matrix with, say, multiple grades of HPMC, you can do that as the weighted average power law model. In this case, we're just using one polymer, so that isn't going to come into play here. The important variables to note in this equation are the calibration constant, which is going to determine the active release from the matrix, the surface area of the matrix, and that's determined by the dimensions of the tablet. It's a 1 gram tablet, so we've defined the dimensions as such. The mass transfer coefficient, which is again based on the hydrodynamics in the system, which we already talked about, the porosity of the tablet, the solubility of the active, and the power law constant, N. The power law constant, N, is an important parameter. It determines whether or not your formulation is, pure diffusion from a sphere, sort of like the Higuchi model. If the power of N is 0.5, that indicates a diffusional mechanism, and as such, then this equation becomes based on a theoretical solution of the partial differential equations of diffusion from a non-swellable matrix. In this case, Corsimire pepis modified this for formulations that are swellable and erodible, N becomes greater than 0.5, and it's typically in the range of 0.6 to 1 or so. In this case, we'll see that N is going to be about 0.78 or so for our formulation, which indicates it's a swellable polymer matrix. And of course, it was an HPMC matrix tablet, so that is obviously correct. The model employs an important physical property of the polymer disentanglement concentration. So that's the concentration at which the polymer is going to detach from the gelled matrix and dissolve into the fluid. And this is based on molecular weight. So as the molecular weight increases, the polymer is less likely to detach from a matrix, and so that's shown here on the right in this plot. So that's also built into the DDDPlus model for CR matrices. So we then built the DDDPlus model for the in vitro release. And here we took the in vitro conditions from the literature publication, which was a two-stage dissolution test where they did 500 mils in HCl followed by 900 mils in phosphate buffer. So we set that up within DDDPlus and set up all the ingredient amounts from that table that I showed in the previous slide. Once we did that, we trained the model on the slightly fast formulation. So we used the optimization module within DDDPlus to calculate what the release exponent and calibration constant were for the API and the polymer matrix. Again, shown here in the equation below. We obtained these values and this fit to the experimental data. So now once we have the in vitro model built, then we can ask the what if question for the formulation we're trying to design. So this is the in vitro data that we had predicted from the IVIVC. And now what I'm doing is modifying the amount of HPMC and lactose in the tablet. The tablet is a 1 gram tablet, so I have to basically co-optimize the amount of filler and the amount of polymer in the tablet such that I get an optimum release rate to match that predicted in vitro profile. So here I've used the F2 statistic in a manual parameter sensitivity analysis. I set up formulations from 300 to 160 mgs of HPMC with a reticent amount of lactose. I predicted this similarity factor and found that 160 mgs was the optimal amount of HPMC that predicted the in vitro release for the fast PK profile target. The prediction is shown on the right for the dosage form. So now the question is, was that prediction the same as what was actually used in the study? Was our prediction accurate? And remember, we didn't know, we're assuming we didn't know anything about the formulation to start with. All we had was a PK profile and 1 or 2 other formulations to generate an IVIVC. So in doing so, we predicted 16 weight percent HPMC in a 1 gram tablet as compared to the actual formulation of 20%. So that's fairly reasonable, just given it's a complete a priori prediction, not knowing anything about the fast formulation, just doing a design of a formulation from scratch. So we used a limited set of in vivo data. We used the IVIVC and GastroPlus, and then we were able to really go in and look at the amounts of the materials in the tablet and design a formulation to hit that PK profile target. So now that we've designed the formulation and we've hit, our PK profile target, we want to see what's the effect on our dissolution specifications or variability in our experiment and or content uniformity on our formulation, and how would that affect bioequivalence predicted in GastroPlus. So in this case, we used a virtual trials functionality within DDDPlus to define a few parameters to do a virtual trial against and see what the variability is. So up here on the top right, we selected the amounts of all the ingredients in a tablet and assumed a CV of 3% for the content uniformity around the mean value. We also assume that the pH of the different phases may vary slightly based on experimental variability. And then also the matrix dimensions may change slightly because of the content uniformity in the system. So we selected all those variables to do a virtual trial around, and we simulated a batch of 25 virtual tablets, and we obtained the results on the bottom right, where we have a high limit of the dissolution rate and a low limit of the dissolution rate and a mean. We can then use these dissolution profiles and import them into GastroPlus to run virtual bioequivalent studies to determine would the expected variability on the low end be bioequivalent with the mean, or would the high end of the dissolution rate be bioequivalent with the mean, or would the high end of the dissolution rate be bioequivalent with the low end of the dissolution range? We can run all those what-if scenarios within GastroPlus. Also, this is, again, using a new functionality within DDDPlus, which is exporting simulated data to GastroPlus files so that you can directly import these into GastroPlus and run simulations. So I used the IVIVC that we developed to convert the in vitro dissolution rate from our DDDPlus model to an in vivo dissolution rate profile using the IVIVC. Then once I imported the imported the in vitro dissolution rate and converted it to the in vivo dissolution rate, I used a Weibull function to fit these dissolution rates, and then just simply used a Weibull function to describe the release within GastroPlus to define the PK profiles using the mechanistic absorption model. Okay, so not only are we going to be using the mechanistic absorption model to predict the PK, but we're also going to be using the population simulator. The population simulator enables you to simulate a population of up to 2,500 subjects. Populations are generated, not sampled from a database, so an infinite number of populations is possible within GastroPlus. Subjects are generated by Monte Carlo sampling of selected parameters with their defined distribution. So any gut physiological parameters, say surface areas, lengths, volumes, we can add stochastic variability to predict what the effect on the PK is. Pharmacokinetic parameters, say that if you were doing a pharmacokinetic model, let's say just the volume of distribution, clearance, that kind of thing. In this case, we're using a PBPK model, so we're looking at the variability in the tissues themselves, the sizes of the tissues, etc. The dosage form and compound parameters, so what's the variability and dissolution rate? In this case, we've already defined what our variability and dissolution rate is, so I'm not actually doing any population simulations based on the dosage form. I'm just using the ranges that we've already ascribed. Populations can be saved and reused for crossover studies, and that is what I did in this case. So I used the same population and tested these virtual tablets on the same populations to see what the effect of bioequivalence would be. Covariation of the model parameters is a complex issue. GastroPlus 9 has some covariate models for the PBPK. However, the results may be slightly pessimistic. Simulated population variances may in some cases be too high or too low depending on what your population is like. So you may have to do some fine tuning of the CVs or the distributions that you use. But it's a conservative approach in many cases. It does, fairly accurately reproduce the variability you see in PK data. So on to the virtual BE calculations. I took those DDDPlus simulations on the left. In this case, the reference is the green, the mean profile of the in vitro dissolution rate. And then the test product is going to be the low end of the dissolution rate spectrum, or the low limit, what I'm calling the low limit of the dissolution range. I've read those in vitro profiles, converted them to in vivo dissolution rates, and ran the GastroPlus simulation in the population simulator and obtained the results on the right that show that the test and reference in this case is bioequivalent. you can see that the Cmax has dropped, and the Tmax is actually quite different in these formulations, but the AUC is fairly equivalent. The confidence interval for Cmax is 80.8 to 95.3, so it is, very close to the bioequivalence limit. So this may indicate to you that you have a perfect specification, or you'd be worried and say that you may want to tighten the standards of your dissolution test, less variability in the method, and then less content uniformity. This simulation tells you, wow, I'm pretty close to the bioequivalence limit. I certainly don't want to get any more variability in the system, and I may want to tighten up my dissolution specifications and then retest this bioequivalence after I do so. We also did the same simulation, but this time the test product is the high limit of the dissolution curve shown on the left, and then the reference is the mean again. And then there's very little difference here. The Tmax is again fairly different, but overall still roughly the same amount of drug ends up entering the systemic circulation, and the AUCs are fairly similar. The Cmaxs are also fairly similar, a little bit higher as you'd expect for the higher dissolution rate, but well within bioequivalence. So there's really not an issue for on the high end of the dissolution limit range in this case, other than the Tmax is quite different. And then we can look at the test being the high limit of the dissolution range and the reference being the low limit of the dissolution range, and then doing the virtual bioequivalence simulation for this case. In this case, we see very similar results to the reference being the low limit and the test being the mean dissolution profile. We see very similar results showing that we're very close to the bioequivalence limit for Cmax and AUC, or for Cmax, sorry, not AUC. So in this case, you would definitely maybe want to tighten your specifications a little bit. So, this example shows the synergy with that DDDPlus has with GastroPlus. It enables you to rapidly investigate the candidate dosage forms that would meet a specified in vitro dissolution time profile that describes an in vivo plasma concentration profile using the GastroPlus IVIVC module. So if you have a target PK endpoint, you can use the IVIVC module to predict the in vivo dissolution rate, and then use DDDPlus to define the content of your tablet to hit that in vitro dissolution. This enables simulation of virtual bioequivalence using the DDDPlus virtual trial capability in conjunction with the IVIVC Plus module on population simulator. In conclusion, DDDPlus is a scientifically based program that uses state-of-the-art numerical methods to simulate in vitro disintegration, erosion, dissolution, effect of surfactants, et cetera, provides insight into interactions amongst the phenomena that govern in vitro dissolution. I hope we've shown that it can help reduce the number of in vitro experiments required to achieve a particular in vitro dissolution time goal and to meet some target PK endpoint. I mean, really, we used only 1 or 2 formulations to then back predict, a formulation to hit a target PK profile, and we did a fairly reasonable job. So these 2 tools can assist with the implementation of quality by design principles and reducing the number of experiments. With that, I'd like to thank everybody here in the Simulation Technologies team. Of course, Vera is our team leader, and then my other coworkers in the team, Haiying, Kay, and Jessica Spires. Of course, we have tons of support from our chief scientist, Mike Bolger, and then John DeBella, who is responsible for a lot of the code in DDDPlus as well. As he used to be a developer, he's now the vice president of marketing, but he's had a big hand in development of this software as well. And all the other teams at Simulations Plus. Obviously, the AdMet Chem Informatics team, who provided the AdMet Predictor module and the QSAR modeling for this project. And then our other groups, computational technologies, discovery, chem informatics, and consulting services. So I think that's all I wanted to go through here today. And so I'll pass it over to John to, start with the questions. And yeah, it was good talking to you all. And
okay. Thanks a lot, Jim. That was a really insightful discussion. And I think especially with that case study that highlights the use of the two software tools together for slightly better in vitro and vivo extrapolation of dissolution principles when we're going into GastroPlus, and then also being able to use DDDPlus to help design or optimize these formulations, and hopefully, as you mentioned, a fewer number of iterations. Now we would like to open it up to questions. There are a few questions that have come in. Once again, as a reminder, if you'd like to ask anything, be sure to write it into the questions pane on your GoToWebinar panel. A couple of questions have actually come in regarding the use of DDDPlus for non-oral products. So one of the first ones is, can DDDPlus be used for topical dosage forms?
yeah, currently, no. We don't have any, like if we're talking about dermal, that kind of thing, we don't have that available within GastroPlus. We are developing certain, other non-oral formulations, controlled release microspheres for, long-acting injectables, that kind of thing. And we're interested in what you guys have to say and what you guys want in the software. Obviously, if you have topical products, other types of products, if we have the data and there's a big enough need for it, I mean, I would be glad to add them to the software and, provide the equations and the tools to do so. But right now we just have the mostly, all the oral products currently.
Okay. Thank you, Jim. And actually, then that second question was related to the ability of DDDPlus to simulate long-acting injectable products. So do you have an estimate on when that might be available for people to use?
Yeah, I mean, it's probably going to be the next version. We do have a couple different working models in-house to predict the dissolution rate of long-acting injectables. One of which has been programmed into our development version here at GastroPlus. We already have with DDDPlus. So we already have a version that contains models that are working, and we're using them internally now. It's going to take a bit more time. We're still adding physics to these models and more complex physics, and trying to really hone in on some of the aspects of the degradation of the polymer, diffusion through the microspheres, the porosity and whatnot. So like I said, we do have working versions of a couple different models in-house, and I would expect that in the next version, we would be able to provide that model to the users.
Okay, thank you, Jim. Another question's come in. Can DDDPlus simulate the effect of ER coating thickness or coat buildup for polymers like ethyl cellulose?
So currently our coated tablet model is it's mainly focused on soluble coatings. I mean, you can put insoluble coatings on there. Whatever coating weight you apply to the tablet, it automatically calculates the thickness of the coating on your tablet. So whatever your formulation is, whatever your coating weight is that you put on the tablet, we're going to automatically calculate what the thickness of that layer is. So in that sense, it's already built in and included what the thickness of the coating is. It's already automatically calculated. Obviously, we're not doing any calculations of predicting that thickness in any tablet coater or whatever, anything like that. We're just modeling the diffusion out of the through the coating. So. But yeah, the thickness is automatically calculated based on your formulation and your coating weight that you apply in the formulation. So it is accounting for the thickness of that coating, and as it dissolves, it's accounting for the thickness as it decreases, eventually reaching 0, and then the tablet becomes an IR tablet. So if you have the solubility values for your coating, it'll, you know, happily model the decrease in thickness of that coating over time, and then the initial thickness based on the mass of the excipient that you added to the formulation.
Okay, thank you, Jim. I'm hoping you know what this stands for, but another question has come in. How do you manage the dissolution from ASD formulations?
I think I know what this is, yes. Artificial stomach duodenum, I think, is the question. We don't have an ASD model currently in DDDPlus. I think, you know, I've seen a lot of a couple different companies give presentations on this, you know, at conferences, and it's definitely, you can sort of mimic these things with phases, but it's not exactly correct because we don't have transit times and whatnot between the phases. So, I mean, this is a good point. This is something that we could be adding in the future to GastroPlus or DDDPlus as a feature. And I think, you know, as more people are using it, and more people are asking for it, it's something we could add to DDDPlus. It isn't currently now in the software. The ASD is currently not within DDDPlus.
Okay. Actually, then there was a clarifying comment that just came in. So ASD in this case stands for amorphous solid dispersions.
Oh, okay. Yeah, so currently most of this is focused on, you know, crystalline drug products. You know, we're going to be, you know, and I think this is an undertaking for GastroPlus too, you know, looking at more amorphous solubilities and the difference between the amorphous solubility and the crystalline solubility, and looking at those dissolution rates separately, and the precipitation rate. It's currently, you know, not specifically built into our models. Obviously, you can always adjust the solubilities and whatnot within DDDPlus for amorphous powders and whatnot, but you know, we don't have a solid dispersion formulation model yet within DDDPlus, but that's a good point. It would be another prime candidate for a future expansion, and I think it is on the high list of things that we're going to be adding in future versions.
Okay, thank you, Jim. Another question, in multi-particulate systems, can DDDPlus simulate dissolution with pH modifiers added in the intermediate coat layers? Currently, no. We currently only have the IR formulation. We are going to be, and we have internally been working on a coated bead formulation for multi-particulates that will account for diffusion through the coating, porosity of the coating, and those different aspects. We were accounting for the osmotic, you know, pressure and the diffusion through those coatings, but you know, pH modifiers is something we could, you know, look at adding to it. You know, it's not released yet, so we still have the opportunity to add physics to our CR model. So I'll definitely look into adding that. But we do have internally a working CR multi-particulate model. It's just not at a level yet which we felt comfortable releasing it. So in the next version, we'll have a multi-particulate model for CR coated bead formulations. So until then, no, but that's on the horizon as well. Perfect. Thank you, Jim. Another question, and this is related to different formulations. Does DDDPlus have the capability to model lipid-based formulations?
Currently, no. We don't have any lipid, you know, we don't have any lipid release models or digestion models within DDDPlus. Again, it's something that we could look at in the future. Again, this is a much more complicated system than, you know, the standard bread and butter dissolution type experiment. So we would really need to look into the physics behind that. And, you know, it's definitely something that would be, you know, a valid addition So we'll add that to our list, unending list of things to do.
Okay, very good. This might actually be a question for Vieira. One or more of the IVIVCs that you showed seem to be more sigmoidal in nature than quadratic. Does GastroPlus include sigmoidal models as well as quadratic and polynomial models? Let me unmute Vieira.
Sorry. Yeah. Not currently. Right now, the models that are built into the IVIVC that allow you to create the correlation between your in vitro and in vivo are limited to linear, power function, and polynomial functions, quadratic and cubic equation. The cubic equation does, in some cases, allow to approximate the sigmoidal shape, so it would allow created an initial lag time, but a sigmoidal model is directly not implemented as a correlation equation.
Okay, thank you, Vieira. This is an interesting question. Is it planned to implement the Z-factor fitting in DDDPlus as it is already available in GastroPlus?
Yes, that's a simple answer. Yeah, I don't really have anything more to add to that other than, yeah, we plan on adding it, and it's on probably one of the highest on the list of additions that we're definitely going to be adding in the next version.
You know, I apologize, but I can't help myself here. If you do take a closer look at the models that we have implemented in DDDPlus, we actually do have what we call a calibration constant, separate from the dissolution rate constant term that we're all traditionally familiar with. And so we kind of think of the DDDPlus implementation to be almost a next generation Z-factor approach, because you can fit a calibration constant to get a dissolution rate, but you could still mechanistically assess what would be the impact of changes to particle size distribution, changes to diffusion coefficient, and so on. And, you know, Jim is showing here in the equation. So this is the mass transfer model. You can see the gamma term, the calibration constant. For those of you who do have experience with the Z-factor approach, you know that what you're doing is you're fitting that entire first term as the Z-factor. And in DDDPlus, what we've got instead are the traditional diffusion coefficient, mass transfer coefficient terms, diffusion layer thickness, particle size distribution separated from the calibration constant that can be optimized. So in a way, there is already a form of the Z-factor model in DDDPlus. And as Jim mentioned, I think we'll introduce the strict definition that is inside the GastroPlus program soon. Another question, can you include a model that allows one to enter tablet hardness rather than compression force?
Yeah, so currently the way you would, I mean, if you have the ability to measure tablet hardness, you also have the ability to measure tensile strength, most likely. So that is the new addition we've added to this version. So if you have a tensile strength value, which you can use a hardness to generate that value, or the same tester can generally calculate the tensile strength of the tablet as well. If you have that value, you can use that Reishkovich-Duckworth equation to calculate the porosity of the tablet by making some assumptions about the bonding constant and the tensile strength of your pure component ingredients. We've added those constants within, you know, the default database of ingredients. So for common excipients like lactose, microcrystalline cellulose and stuff, those are already defined, and you can do, if you just have a tablet tensile strength value, you can predict porosity based on that. So that's one thing we've added to this. So it would be coming in as tablet tensile strength, but you should be able to get that number if you're able to generate a tablet hardness as well, I would think.
Okay. Thank you, Jim. Another question, are there any thoughts of being able to input the API polymorphic form into the DDDPlus model?
Yeah, I mean, currently we have salt forms, where you could maybe account for your polymorph is in the solubility if there's some differences. Maybe if you have a certain polymorph. We don't specifically account for multiple different polymorphs of a drug substance currently. I mean, you'd be using the input for whatever the main polymorph of your compound was within DDDPlus. So, I mean, specifically, you could add inputs that approximated whatever polymorph you're using, but we don't allow multiple inputs for multiple different polymorphs, I guess would be my answer to that. And I don't know, that would be I guess on another thing on the large list of things to add potentially if at some point, but I think the amorphous solubility would probably be maybe the first thing we'd go for, but again, hopefully that answers the question there.
Thank you, Jim. Related to, I think, some additional feedback for potential new features and upcoming versions, are there plans to include CFDs associated with peak vessels into DDDPlus?
Peak vessels. So, I mean, I can tell you the only thing we have planned on right now, as far as vessels are concerned, is maybe adding some of these other small-scale dissolution vessels, like maybe the Sirius instrument. Some of these vessels that do the permeation with dissolution. So PION has one, and also Sirius have, they're where they put an oil layer on top of, or an octanol layer on top of the two, aqueous layers and do the dissolution there, so that drug can be extracted. Those were the main apparatus that we're thinking of adding currently. Those are on the horizon. We haven't thought about adding peak vessels, CFD, but
Thank you, Jim. Another question here is what simulations, dissolution simulations in DDDPlus account for excipients like lactose or methylcellulose, and are these excipients accounted for in terms of interactions with the drug in solution after dissolving?
Yeah, that's a good question. And yes, I mean, we did, as I said, when I went through the short, couple slides on the equations, I mentioned that we have models to predict interactions between the excipients and the API. So we have models to predict the effect of wetting agents, solubilizers, disintegrants, whatnot in the models. So in terms of solubilizers and surfactants and whatnot, if you know the solubility of the drug in these and solutions of these excipients, basically you can calculate the partition coefficient, or the K value that relates the concentration of the solubilizer to the enhancement and solubility. In DDDPlus, it's accounted for as the solubilizer dissolves, and again, we're using the mass transfer calculation or equations to calculate the dissolution of the excipients based on, you define the particle size of all your excipients. You have to know what the solubility of your excipients are, the pH solubility profile. So if you have really good data on that, you just input that in there, and we're going to use the same mass transfer equations. So as the solubilizer, let's say, dissolves and that gets into solution, it's going to enhance the solubility of the active, and we account for that as it's dissolving. So as more solubilizer enters solution, the active concentration or solubility in the media is going to increase. And And as far as wetting agents, as wetting agents dissolve, it's going to have an effect on how fast the API particles dissolve in the other excipients. So we are accounting for the concentration of those different excipients and dynamically calculating their effect on the dissolution rate of the active. So it's a good question, and I didn't go through those examples, but that has been part of DDDPlus for a while now. So if you have solubilizers, let's say, PVP or something, or some sort of maybe PEG or something in your formulation that enhances the solubility of your active, that's going to be calculated as long as you have some in vitro data to define the K value, or you can optimize the effect on the active as well if you just have experimental data of drug release. So, but these are calculated dynamically, and full dissolution model for each excipient is provided.
Okay, thank you, Jim. To try and get things wrapped up here within the next few minutes, I'm going to combine two questions here that were asked. And this one, again, might be for Vieira. Does the IVIVC approach also work for animals? And is there any experience potentially extrapolating an IVIVC built with preclinical data to a clinical PK prediction?
Well, the answer to the first question, I don't see why it wouldn't work for animals, right? The goal of IVIVC, especially when using the mechanistic model, as Jim was explaining, is to correlate the dissolution happening in vitro, or the dissolution that you measured in vitro, with the deconvoluted dissolution in vivo. So you are trying to, the correlation would basically be accounting for the differences in dissolution rate caused by the differences in environment, in your in vitro versus in vivo environment. So there is no reason why, especially the mechanistic model, why you should not be able to use or to build IVIVC for animals as well. And GastroPlus, as you know, does include the physiological models for all the common preclinical species. That being said, as to whether the IVIVC would extrapolate from animals to human, we haven't tried that kind of work yet, based on, again, what the IVIVC is basically accounting for. I would not expect that, because, again, you are creating equation that basically you are relating the differences in the in vitro dissolution to the in vivo dissolution. So if you are getting anything but the one-to-one, you are basically looking, your equation would be accounting for differences in the environments in which the drug is dissolving. And again, in human, the environment would yet be different than in mouse, rat, and dog. So unless the differences in dissolution are caused by anything else than the solubility of the compound is affected by differences in pH or bile salts, any other effects, might not translate between animals to humans. So you'd probably be developing a different correlation for human, different IVIVC.
Thank you, Vera. Question about DDDPlus. Is it capable of modeling systems that exhibit supersaturation and precipitation?
Yeah, so I think we talked about that. If you use the, well, for amorphous, I already talked about that situation. Now, if you are talking about a two-phase dissolution type experiment where you're looking at gastric to intestinal buffer transfer of, let's say, a weekly basic drug that's going to, completely dissolve in gastric buffer and then be transferred to a higher pH, we can account for supersaturation and precipitation within the DDDPlus models. So we do have precipitation that's included with the models. So in that case, yes, we can account for it. As was mentioned in the case earlier, not for amorphous yet, but I think we'll be getting there soon enough. So
okay, very good. We'll wrap up here with two more questions. The first, are there plans to incorporate dissolution of granules, granulations as a function of manufacturing and physicochemical properties into DDDPlus?
Yeah, I knew we would get this question. This always comes up. So yeah, right now it's just tablet to primary particles. We don't have a granule model. Granulation is so dependent on process parameters, composition, roller. I mean, let's say you're doing roller compaction and that type. So dependent on processing parameters. I think it would be difficult to get a mechanistic model for all these different situations. But I think the ability to add dissolution of granules is important because sometimes it can be the limiting process in dissolution. I mean, not usually for an IR tablet. I mean, most times you can design a granule that's going to release the API and not ever be the limiting mechanism. But it does come up sometimes, and so at least having an option of simulating those, even if maybe it's more semi-mechanistic at first, I think would be a good addition. And we're thinking about that as well. So that's a good question. And I mean, obviously, if anybody has lots of data on granulation and processing data, we would love to build a model for it. So I mean, we always would like to work with anybody as well to define these models and add them to our software as well. So it's a good question.
Thank you, Jim. And we'll wrap up with this question, probably for both of you. Do you foresee the DDDPlus simulations being utilized as a justification to the agencies for dissolution waiver for commercial batch releases?
I would hope so. I think it's a good, at least a good set of information, just even if it's not used for any sort of approval purposes, just to show that you understand your system and, can simulate what's going on. I think it would be, I hope that's the way things are going. I don't know if Vera has a comment, but.
Yeah, I don't have any other comments to that. I would expect that if that the acceptance would be increasing as the time goes. We know that the agencies are very much in favor of increasing the use of modeling and simulation in different submissions, and we expect that as everybody, including the agencies, are increasing their experience in even in vitro modeling, that this would be increasing. But the proof is in the pudding, so we'll see how it goes. But I would expect that it could be used.
Okay, thank you both. There are a couple of more questions regarding pricing, licensing, evaluation requests. I just want to clarify that you will be receiving an email within the next day or so, thanking you for attending today's webinar, and it will include some instructions for requests of any information that you might need from us. So please keep your eye open for that email coming through. You'll have different email addresses in that message that you can utilize to reach back out to us for copies of the slides, evaluation requests, or pricing information. And so with that, we're going to conclude today's very excellent webinar. I want to again thank Jim and also Viera for the insightful presentations and also the Q&A session. If you did miss any part of today's presentation and you're interested in the playback, it should be available at our website within the next day at www.simulations-plus.com. Thank you again for taking time out of your schedules to learn more about DDDPlus and GastroPlus, and have a wonderful day.